Well, what I'm going to talk about now is the combined activity that occurs on these three different particular organs and how it's essential for digestion. Okay, so now, first thing we need to talk about is the structure of the pancreas. So I'm going to try to draw the pancreas. It tends to be really easy. Right? Anatomically, that's what you see, right? It's just this long thing that sits kind of partially into the stomach back there. So the pancreas has two functions. One is called the endocrine function. And the endocrine function of the pancreas is something we're going to be talking about in a couple of weeks, because the endocrine function is going to be all about insulin and glucagon. And by the time we're done with the metabolism portion of this block, you are going to be absolutely sick of me talking about insulin and glucagon. So I'm going to save that for later. What we're going to talk about now is the exocrine function. So remember the difference between endocrine and exocrine. Exocrine is when you have a secretory function that is going to be putting hormones or other factors into duct systems that are going to be expressed outside the body. Outside the body in this case means in the lumen of the GI tract. Endocrine function means a secretory function, putting hormones into the blood system that's going to circulate inside the body. So the exocrine function of the pancreas is going to involve acinar units that are structured the same as what we saw in the salivary glands. So we are going to have the secretory epithelial cells lining this dead-ended cul-de-sac, and they are going to be emptying into the lumen of these acinar units, which will drain into these small little channels and ducts in the pancreas that will eventually all lead into what is called the pancreatic duct. Okay? Now, that defines the anatomical structure, histological structure of the exocrine pancreas, the acinar units and the intercalated duct system. The endocrine units, I think most of you probably know what these are. These are the little islets, islets of Langerhans. And these are where the alpha, beta, and delta cells are that are going to be controlling the insulin and glucagon activity. So we'll get to those later. So where I want to join these three different organs together is at the very end of the pancreatic duct. Now the pancreatic duct is going to be joining another duct. And that is going to be coming down from the gallbladder. And that, of course, is called the common bile duct. Now, at this juncture, I like to call it the confluence, because it's like two rivers coming together. This is called the ampulla of Vater, also called the hepatic or hepatopancreatic duct, because it's the joining of the drainage from the common bile duct and the pancreatic duct. Now, ampulla, remember what ampulla means? It's like a vestibule. Oh, you're all too young, that's right. In old times when you have a house built, you open the front door and you don't enter the living room typically like most of our houses do today. You enter a vestibule, and that vestibule can be actually walled off by another door, and there's like it's like a little mud room kind of thing, and that's called a vestibule. And the ampulla is the same thing. So it's the vestibule, the joining center of the common bile duct and the pancreatic duct. Now, this is going to empty into the duodenum. We need to be able to control all fluid movement in the body under all conditions. And how do we do that? We put in a sphincter. And this is the sphincter of Oddi, not the sphincter of Garfield. Did you get that? Okay. You'll never forget this. Some things you can never unlearn, right? So the sphincter of Oddi is going to be controlling the outflow of material from the gallbladder and the pancreatic duct. Now, the reason I bring these organs together is because there's a lot of coordinated activity in the physiological regulation between these organs, the gallbladder and the pancreas. Okay, so that's the basic structure we're going to talk about. So it's this area right in here, the ampulla of Vater and the sphincter of Oddi, is where I want you to focus your attention for this whole hour. Because we're going to talk about how material moves in and out of here, and then we're going to start talking about what the components are of the material that is coming out into the duodenum. Okay, so we look at a typical acinar unit. In the exocrine pancreas, we see our epithelial cells, and these are the secretory epithelial cells. And these particular cells are going to be performing the same type of function as a salivary gland. So there's nothing different about this. The components, of course, are different. So on the outside of this acinar unit, you have, just like the salivary glands, myoepithelial cells. And these have a contractile function. So you can imagine that we're going to be able to stimulate this so that we can squeeze that acinar unit and eject the material into the duct at the appropriate time. Now, these particular cells in the ducts themselves are also going to have a secretory function. Their secretory function is going to be the exact same function as the ductal cells in the salivary gland. They are going to be secreting bicarbonate ion. They are also going to be secreting a very large volume of water. Now, the purpose of this, the bicarb again is a base. The function of this particular base is to neutralize the gastric acid. So as these materials coming out through the pyloric sphincter, it still is a pH of approximately 2 to 3. Now the duodenum does not have the protective function that the gastric epithelium provides. It has some mucus glands, but it's not nearly providing that sort of protection against acid. And so this is why duodenal ulcers can be so common. Because if you don't have proper protective function by this bicarbonate secretion, this acid erosion is going to keep slamming into that duodenal wall. So that one description that was made earlier in the blog talking about the duodenal ulcer being on the proximal wall. So if you have the way this is coming out, I don't have this drawn correctly. So if this is the pyloric sphincter, the duodenal bulb comes out here. The most common place for a duodenal ulcer is that opposing wall. And the reason for that is because the force of the material coming through that pyloric sphincter is going to be slamming against that wall. And so this is the highest level of acid exposure, and so that's the big area that ulcers can form. Now, we've got the bicarb coming out to neutralize the acid. The huge volume of water that also is secreted into these ducts that is going to come down the pancreatic duct is extremely important because of the components that are being secreted by the acinar cells. What's in this lumen is the major portion of the enzymes that we need for digestion. So these cells will be making proteases. We'll talk about these next week. These proteases are involved in digesting proteins. So this automatically tells you that the pepsin that is located in the gastric lumen that is activated from pepsinogen because of the acid, that is only the very beginning of protein digestion. And it's actually quantitatively serving a minor function. The series of proteases that the pancreas makes, that we'll talk about all of these involved next week, this is the major digestive portion for our proteins. We also have a lot of lipases, pancreatic lipase. Now, I'll make a note on here that in some of your readings, you'll notice that there's also lingual lipase. Lingual lipase is one of the components in salivary secretions we didn't talk about because it contributes almost nothing to the adult diet. There's also gastric lipase that's being synthesized and secreted by chief cells. I didn't talk about that because that contributes almost nothing to adult diets. The pancreatic lipase, this is the big one for triglyceride digestion. And triglycerides are the major source of our fat in our diet. Now, I want to put a little star on this, a little corollary, that in newborns, if the pancreas is not fully developed, what does that tell you about lingual lipase importance for a newborn? It's very important, yeah. So lingual lipase in a newborn is critical. As we get older, as the pancreas matures, now we are producing the pancreatic lipase, and the lingual lipase in an adult really doesn't contribute much of anything. I'd put a little star on there because that's one of those things that board questions could ask you, because we are trained to think that the pancreatic enzymes are the major digestive enzymes, and they are, with certain exceptions. So the other enzymes we have are amylase. And amylase, of course, is our carbohydrate digestion, primarily starch. So the contributions of amylase in our salivary gland to carbohydrate digestion, again, is minimal to non-essential, really, because the amount of amylase being produced in the pancreas is the major force for digesting our carbohydrates. And not to leave them out are nucleases. And nucleases, of course, are going to be digesting DNA and RNA. We do have DNA and RNA in our food because if we're eating any kind of life material, plants or animals, there's going to be DNA and RNA. And what we're able to do with these nucleases is digest them down into their base structures, and then we can absorb the bases and use them to our own advantage. Now, in textbooks, physiology textbooks, nucleases are not addressed very much in medicine, pancreatic nuclease. And the reason for that is because proteases, lipases, and amylases are digesting the major portion of our food products and providing us with energy molecules and building blocks, the major ones. Nucleases, not that important because we can make our own bases for DNA and RNA synthesis, and even more important is that we have what's called a salvage pathway, that we can recycle our bases so that we don't have to make them. So once we make them, we can recycle them in our body and continue to use them. We'll get into that in about 3 weeks. Okay, so look at these here, proteases, lipases, amylases, being secreted into the ducts. Are these components, these digestive enzymes, something that you want to hang around in your ducts? If they do hang around, what could they possibly do pathologically? Self-digestion, autodigestion. So we're going to talk next week that these are actually, many of these are secreted in a zymogen form. They're inactive. But there's also a process called autoactivation. and that takes time. So if these are secreted into the pancreatic ducts and they hang around a little too long, you can get autoactivation of proteases which can digest your pancreatic cells, causing pancreatic insufficiency. We'll talk about that in a few minutes. But it's the secretion of these ductal cells that put out this bicarb and put out this high volume of water that is flushing the ducts. And so that's the most important feature of this water going in. And it literally is 1 to 2 liters. It's a huge amount of water. And that's just to flush these enzymes out of the pancreas and into the duodenum where they're going to do their most good. Okay, so now how is all this regulated? I've given you a couple of pictures because there's 2 types of regulation for the pancreatic secretions. One is going to happen in the cephalic phase. The other one's going to be in the next phase that we haven't gotten to yet called the intestinal phase. So let's boil this down to the most important elements. So this is the apical surface. So this means this is the lumen of the acinar. So the apical surface, basal surface. So the basal surface is going to have all of the regulatory elements. And now let's talk about the cephalic phase. And the purpose of the cephalic phase is exactly what we've seen in the salivary gland regulation and the gastric gland regulation. Cephalic phase, smelling, tasting, seeing food. Our CNS centers are sending signals down through the vagus, and this is going to innervate through acetylcholine and M3 receptors. And this is going to trigger, what's our mantra? What's pathway? PLC, IP3, calcium. And when we do that, this is going to induce the translocation of these vesicles to the surface of the pancreatic acinar epithelial cells. Now, in these vesicles are our digestive enzymes. Now again, it's the same principle that we're going to see pretty much throughout the entire GI tract. We need instantaneous responses to our dietary components and consumption. So we do not have time in our pancreas to make the enzymes de novo, because that can take 15-20 hours. So we are going to pre-synthesize these and store them in vesicles. And so all we need to do is provide a signal for fusing these with the membrane and then secreting these into the lumen. Now the cephalic phase, again, the same purpose of the other glands, is to prime the pump. We're preparing ourselves. So now here, we're going to start secreting a little bit of our digestive enzymes into the pancreas, preparing for the way the food's coming down. Now the other signaling that you see in here is through GRP. And GRP, that's gastrin-releasing peptide, that also plays a role, but it's a minor role. The big role is the acetylcholine M3 response. Okay, so that's the big one that shows up in all the textbooks. Now that's the cephalic phase. Now, the intestinal phase begins the same type of principle as when the gastric phase begins, when food enters that particular location. So the gastric phase was initiated when our dietary molecules entered the stomach, and those dietary molecules interacted with the G cell. By doing that, it secreted gastrin, and then we increased our possibility with the hormonal signaling to start producing acid, and that gave us the big gastric phase. The intestinal phase, same principle. When dietary molecules come out of the gastric lumen and into the duodenum, that begins the intestinal phase. And of course, at that point, the dietary components are now in the duodenum, and so we need to get these dietary enzymes as quickly as possible into that mix. So what we're going to have now is a hormonal control. And this is CCK, cholecystokinin. Now, CCK is an absolutely huge and important hormonal regulation in this entire process. And when I mean entire process, again, focus on that ampulla of Vater, that interaction, that intersection there, because CCK is going to activate your pancreas, and it's also going to activate your gallbladder. So CCK is the master hormone that is going to put the pedal to the metal and get everything secreted out of the pancreas as quickly as possible, and also the gallbladder. So CCK is going to feed into this same pathway. And again, what we have here is the vagal stimulation still, and we also now have CCK. So remember, this is potentiation. So now we're getting a synergistic activity so that we can really increase that calcium level to really cause all of our vesicles now to fuse with the membrane. So now we're getting all of these components dumped into the lumen. So how do we get these enzymes out of this acinar structure? The CCK is also going to stimulate the myoepithelial cells. So it's increasing secretion, and it's also activating contraction on the myoepithelial cells. And so now it's a similar kind of activity as the salivary gland. You get that big contraction, and all of these acinar units are just pushing their luminal contents out. Now, we have to look at CCK, because CCK is the master regulator of the intestinal phase. It's the initial opening of the door. And so we'll get to the gallbladder in a second, the effects on that. But where does CCK come from, and how is it triggered? This process of CCK is rather complicated of how it's sensed and how it's secreted. What I'm going to do is give you the gory picture, because I don't know where the step 1 exam is at this point on CCK. The reason I'm cautious about that is because personally I do not believe you need to know all of these gory details to practice good medicine. But this is present in our textbook by Kim Barrett in Gastrointestinal Physiology, and this is an important textbook that is used in all medical schools, and that means step 1 test exam writers, test question writers. So I'm not sure what level of detail that you really need. For this block, I will stick by my philosophy that you don't need all of these gory details. Okay? But just remember, for step 1, I'm not sure where they're at on this. So what we have in the intestinal epithelial cell if this is the lumen of the duodenum, this is the apical portion of the cells. This is the basal portion. So now what we're going to have is another very cleverly named cell, as Dr. McGuire said. It's called an eye cell. Very creative. I don't know who comes up with this stuff. So the eye cell is going to be the cell that is going to be secreting CCK. Now, what I'd like to say is that this process is as simple as gastrin, but it isn't. There is a sensor. That sensor is going to be located on an adjacent cell, and it is going to be detecting primarily peptides and amino acids. It can also detect fatty acids that we are starting to generate from lipase activity. And so this particular cell is going to be secreting a factor that is going to bind to the eye cell. And this particular factor is called CCK releasing peptide. So what CCK releasing peptide is a positive acting factor for the release of CCK. So dietary components, proteins, amino acids, fatty acids, stimulate the release of CCK-RP, and that binds to the eye cell and now tells the eye cell we need to secrete CCK because now we have dietary components in the duodenum. But that's not enough. Now what we have also is another peptide called monitor peptide. And this is coming from the pancreas. That also is going to bind together with the releasing peptide and be a positive catalyst to stimulate the signaling required for CCK. At this point now, we have CCK secretion. Does anybody see a problem with my drawing? I think you're witnessing a senior moment here.
We didn't want to drop it the other direction.
Bingo! Thank you! There's what happens when my head is so focused on the releasing peptide and monitor peptide and I forget, oh yeah, CCK, it's like a hormone, right? So it's got to go into the blood. Otherwise, how would it get to the pancreas? Has everybody got that? Correcting my error there? Senior moments? That's a great excuse when you get older. Okay, so we've got these two factors coming in from two different directions that are going to positively stimulate the CCK secretion. Now, once the CCK is correctly in the bloodstream, not in the lumen of the duodenum, once it's in the bloodstream, now the CCK can interact with the pancreas plus the gallbladder. Now, we all know, because everybody recalls very strongly and excitedly and enthusiastically your biochemistry. Woo-hoo, right? I'm priming you for like a couple of weeks. Whenever you have a positive signal in biochemistry, isn't that dangerous? That's called a positive feedback or positive feed-forward reaction. So that means that you put something on and you accelerate. Why is that dangerous? Everybody's tuning me out exactly right now. I can see it. So when you got this acceleration, you've got something that potentially can go out of control. In biochemistry, we are all control freaks, and everything has to be under control. So once you've got a positive stimulation, you have to have a reverse in some sort of inhibitory signal to put the signal in check. Same principle we saw with gastrin. Gastrin is a beautiful hormone. We need that to get our parietal cells chugging along to make acid. But what happens if gastrin doesn't stop its signaling? What type of tumor is that called? Gastronoma, right. And what happens in gastrinomas? What happens to the patient? Hyperacid secretion, ulcerations, erosions, all kinds of problems. So we need a break on this. We need to put this under check. And the gastric glands, we have talking about somatostatin that puts the stop to this and puts a negative inhibitor to check on it. Now here we have to find a way to turn off CCK secretion because we don't want our secretions coming out of our pancreas and gallbladder all the time. We want it only during meals. So here's where it gets really stupid. Trypsin is the major protease for digestion of proteins that is secreted by the pancreas. Trypsin is going to be digesting dietary proteins. These dietary proteins are going to be occupying the enzyme active site of trypsin a lot because there's a lot of dietary proteins. That means that trypsin has a very difficult time getting to monitor peptide and CCK releasing peptide to digest those and hydrolyze and get rid of those. Which means that the monitor peptide and releasing peptides are present for quite a while, which means that they can continually stimulate CCK secretion. But what happens when you've completed digestion in the proximal duodenum, and now your dietary proteins start to diminish? The trypsin is still there. The active site of trypsin now becomes available because it's not being occupied by our dietary proteins. So now the trypsin is looking for something else to digest, and that's the releasing peptide and monitor peptide. And by digesting those peptides into amino acids, you get rid of them, and so you stop the signaling on the eye cell. So what's happening is the dietary proteins are out competing the regulatory proteins for trypsin activity. When the dietary proteins decrease in amount, then now they're no longer able to outcompete for trypsin binding, and now you degrade your releasing and regulatory peptides. Now what's important to me? What's important to me is that you know that it's dietary components entering the proximal duodenum that triggers release of CCK out of eye cells. Know generally that there's these regulatory proteins that are degraded by trypsin, and that's what is the brake to shut off CCK secretion. Because I think those are very, very important elements to know in medicine. These other details are getting a little bit heavy on the regulatory element because I am not aware of any type of disease state or therapies that are focused on these two particular regulatory peptides. Okay, we're good with that? Now, I haven't talked about bicarb in water. I'm talking about CCK stimulating release and contraction to get all of those digestive enzymes out of the acinar lumen. But what about the bicarb in water? Well, here we have our ductal cells, which are putting in the bicarb and water. Now again, remember, as that gastric chyme is coming out of the lumen through that pyloric sphincter, that pH is still very low. There's a lot of hydrochloric acid in there. We have to protect that duodenal bulb as quickly as possible. So what we're going to do now is we are going to increase the amount of water and increase the amount of bicarb that's secreted by the ductal epithelial cells. This is going to be done by a hormone called secretin. Remember secretin? We talked about that as a test in the gastrinomas. Now, in the gastrinomas, remember this is the opposite. And in the article, you'll see the word paradoxical. And that means opposite of what we anticipate or expect. And so secretin, under normal circumstances, is able to help inhibit gastrin release. So that's a secondary function of secretin. The primary function of secretin is to bind to the ductal epithelial cells in the pancreas and increase the amount of water and bicarb that flows in. So now you've got coordination between CCK contractile induction and inducement of the secretion in the acinar cells, and you've got an enormous amount of bicarb and water pouring into the ducts, so you get literally a fire hose coming out of the pancreas. Now, secretin is responding to acids coming out of the stomach. And so this is done by cleverly labeled as S cells. Now, if S cells are responding to acid, and in response they secrete secretin, what type of cell is an S cell? Open endocrine cell. Yeah. What's another cell type in the gastric lumen that is similar to the S cell? D cells. Yeah. So the D cell and the S cell are performing the same function. The D cell is sensing acidity and it's secreting somatostatin to put a brake on the G cell and the ECL cell. And the S cell is detecting low pH, and in this case, secreting secretin in order to increase bicarb flow to neutralize. Okay, so now I want to take a little bit of a detour here and talk about cystic fibrosis. Because cystic fibrosis, we tend to think about that manifesting as a lung problem. And it does. Cystic fibrosis, the problem there is because of the CFTR, cystic fibrosis transmembrane regulator. That particular transporter is a chloride channel, and with absence of that chloride channel, you get reduced water flow, and so in the lung, that leads to an increase in the viscosity of the mucus. And so that's how we start getting all of those lung issues in cystic fibrosis. But cystic fibrosis was originally discovered in the pancreas. And why is that? Well, we have to talk about that water flow. So the picture I've given you in the notes, this again is from our Kim Barrett text. All of those transporters on the basolateral side, don't worry about those. That's a physiological nightmare, and the reason those are put in there is the same reason I said before, is just to make sure you can see that the ions are balanced going across the plasma membrane. What's important is on the left side and the apical side. So normally, the way water works, the water transfer works, is that the secretin is going to stimulate the cell. And this is going to go through your GS, increase cyclic AMP, increase protein kinase A activity, and then protein kinase A is going to phosphorylate the chloride channel, CFTR. Once this phosphorylation reaction occurs, chloride will now flow out of the cell. If chloride flows out of the cell, that is a negative ion. And remember a couple days ago I said that charges are only maintained in certain areas, functional areas of the body, like for neural conduction. But charge differences are abhorred in the body. So we have to neutralize this charge. So what's going to come out to neutralize this charge? What's the most common positive cation in the body? Sodium. Yeah. So sodium is going to come through either a pericellular, in between cells, or it's going to come through transporters on the cell membrane and apical membrane. Now, remember your osmotic theory. If you got ions flowing in one direction, what chases the ions? Water. Yeah. So because of these ions, now we have the water being pulled in. And it's by this mechanism here is how we get the incredible amount of water from these ductal epithelial cells. Now, the other part of this is the bicarb secretion. And here we have a transporter that is going to be secreting bicarb. And it's an antiporter, so it's going to take advantage of some of these chloride ions that are flowing out to help drive the exit of the bicarb. And we all know where bicarb comes from, right? Because we're all up on our biochemistry. Remember bicarb? Somebody want to volunteer? Give me hope, give me faith.
Carbonic anhydrase.
Carbonic anhydrase. Carbon dioxide from metabolism, primarily, combined with water to make bicarb. Okay? So now, what happens in cystic fibrosis? Well, this chloride channel is not there. Because remember, the primary form of cystic fibrosis is a mutation in the chloride channel in CFTR that prevents it from being fully formed. So it gets translated and then it gets degraded immediately and it never appears on the membrane. So if this happens, you now have dismantled your capacity of pushing water into the lumen of those ducts. So go back to what we talked about initially with the enzymes coming out. What's going to happen to the pancreas under these conditions without that chloride channel? Pancreatitis, yeah. This is also called pancreatic insufficiency. And what's happening in cystic fibrosis is because you've lost this chloride channel, you don't have the water coming in there to wash those enzymes out of the pancreas. And so cystic fibrosis is simply an autodigestion of the pancreas. So if you have autodigestion of the pancreas, what's a clinical symptom you're going to see?
Pain with eating.
Pain with eating. Could there potentially be malnutrition?
Yes.
Big time, yeah, because we're not producing these enzymes. Is there a therapy that you think you could provide somebody for that? Is there something you could give to your patient orally that might help compensate for this? Have you heard about the enzymes that can be packaged into little pills you can take orally? Yeah, digestive enzymes. We can take these orally because we can purify them and put them in purified form in a pill. What's important about this pill? Orally, down the esophagus, into what kind of pH environment? Acidic. What's the only protease we have that can handle pH 2? Pepsin. So what do we have to do to this pill to protect it from that acid environment before those proteases and enzymes get denatured? Coated, yeah. So these pills are coated specifically to be able to withstand that acid environment and then release into the duodenum, into the intestine. Okay? Now, water secretion by the small intestine. So this is the water that comes out of the pancreas to assist in digestion. Remember, digestion, all the digestion enzymes are hydrolytic enzymes, so they all require water for digestion. But the amount of water coming out of the pancreas, even though it's a lot, it's insufficient to be able to supply sufficient water for motility of the small intestine and complete digestion. So that means the small intestine is going to be putting in an enormous liter upon liter upon liter of water into the lumen. Now, how does that work? If we have an intestinal cell, epithelial cell, this is the luminal side of the small intestine. We need a mechanism to trigger water release during digestion. And so in this case, we're going to talk about vasointestinal peptide, VIP. Now we know this is released because this is released, as Dr. Shuttleworth said, this is released in response to digestive signals. We saw that this is a relaxation factor in this LES. This also has another function, and this is going to be binding to the basal surface, and it's going to bind to its receptor, and it's going to signal adenylate cyclase to increase cyclic AMP to increase protein kinase A activity. What the protein kinase A is going to do is the same thing in the ductal cells in the pancreas. This is going to phosphorylate CFTR, the chloride channel, opening the chloride channel to allow chloride ion to flow out. Unfortunately, because of our Western diets, we have plenty of sodium everywhere. The sodium is going to follow that to neutralize the chloride, putting lots of sodium chloride out there, and we are going to get an enormous amount of water pouring out of our tissues into the lumen. Okay, so it's the same principle in both cases, same players that are doing this. And it's just an osmotic pressure that's doing this. Now, something we're going to see next week is the reverse of this. We are putting in liter upon liter upon liter of water. And all of that water is not coming from our diet. It's coming from our tissues. So that means we need to recover this very efficiently. Otherwise, we're going to have diarrhea, which is a horrible thing, but even worse than diarrhea is dehydration.